Now that we have a really clear view of what was going on in the electron transport chain, we can return to our poster, which is ever evolving and hopefully gaining lots and lots of detail. In fact, you're going to want to pull out your poster right now. You're going to want to pull out your poster every single time you do a homework assignment, when you take the exam, when you take the final exam, because it's going to be your resource forever. So let's think about what's happening in the ETC and what kind of details we're adding to our poster. The NADH, which of course is coming from the TCA or potentially from the transition step, is going to get oxidized at complex 1. So complex 1 oxidizes NADH and it becomes NAD+. Once it has lost those 2 electrons, of course, now the NAD+ can go off to pick up more electrons, get reduced. So now complex 1 is reduced and it transfers electrons throughout electron carrying centers within that complex. It utilizes the energy that's released to pump protons. We know that it pumps 4 protons. That contributes to the proton motive force, but electrons are then passed onto a mobile carrier called Q, and when that occurs, Q is converted into QH2, and QH2 can now flit through the membrane, taking its electrons to complex 3. Please try to visualize with me that this is in 3-dimensional space, and the membrane comes out at you and goes into the page. So the QH2, that is a lipid-soluble molecule, can flit around in the phospholipid tails of the phospholipid bilayer, and it can make its way over to complex 3. So QH2 is oxidized at complex 3, converted back to Q, and electrons are passed throughout the electron carrying centers in complex 3. Complex 3 is going to pump 4 protons. Now, of course, from complex 3, cytochrome C is going to pick up electrons. It's going to transfer them to complex 4. And in complex 4, electrons again will get passed through all the electron carrying centers, energy is released, and 2 protons are then pumped. We now recognize that a full 10 protons have been pumped from the energy coming from just 1 NADH and the oxidation and transfer of electrons from 1 NADH molecule. We're also noting that the last thing to pick up the electrons inside of complex 4 is the O2. In an aerobically respiring organism, oxygen is the terminal acceptor. Now, there are organisms out there that don't use oxygen. They use another molecule besides oxygen. And of course, we then know that they're anaerobically respiring because they're not using oxygen. But hey, oxygen is good if you can get it because it's got the highest standard reduction potential. So when it is reduced and water is formed, that is that final product, right, of the oxidation of glucose. Wonderful. So aerobic respiration ends there, except for that we now know that we've got this beautiful PMF that has formed, and we know that 10 protons can swoosh in to the ATP synthase machine. And those 10 protons turn the turbines of the C subunits, which turns the gamma, and gets all of the action going at the catalytic alpha and beta subunits. That allows ADP and inorganic phosphate to be exchanged for ATP, aka this is oxidative phosphorylation. So we're recognizing then that 3 ATPs, right, this would have been the stoichiometry on that for the 10 protons that would be made there. Now that we've gotten a really good look at that and our poster is ever evolving, I want to go ahead and take some time to think about how this would differ in a bacterial cell. So if I didn't bring Norbert home with me today, but I should, like a pet, right? If we had Norbert with us and we wanted to look at how Norbert would be different, that is, where would Norbert's electron transport chain be? Well, unlike this yeast cell, right? This is our yeast cell over here. Unlike our yeast cell, we know that we would see on Norbert that the electron transport chain would be found across the inner membrane. So if we were to look across the inner membrane, and of course you can tell me what that makes Norbert if this is the inner membrane, outer membrane, and the periplasmic space, right? Okay, so this is the complexes, if you will, of the ETC. Now, the thing that's interesting is that these complexes are not in a bacterium, they're not identical typically to what we see in the mitochondrial ETC. Sometimes they're similar, like Paracoccus denitrificans actually has a very similar ETC to the mitochondrial ETC, but in a lot of bacteria, this ETC varies. In fact, it would take us an entire separate course to tell you all the things about the bacterial electron transport chains. And so I know that it sounds like a cop-out to simply say that the components of the electron transport chain in prokaryotes vary widely, but it's a cop-out except for that it would take us an entire semester to learn all about them. So that's why we're not going to go into details on every single one of them. But we will suffice it to say that some bacteria do contain some of the components of the mitochondrial ETC, for example, cytochrome C oxidase. Remember that that was complex 4, the terminal member, the one to which oxygen bound. We can actually assay for the presence of cytochrome C oxidase in the test called the Oxidase Test. So the Oxidase Test is equivalent in the gram-negative world to the catalase test in the gram-positive world. That is, it's very good at distinguishing two major groups of bacteria. This is the test we use to differentiate Pseudomonasaceae from Enterobacteriaceae. So E. coli and Shigella and Salmonella are negative for the presence of cytochrome C oxidase, but Pseudomonas is positive. So we can assay for that. We can also make note that in E. coli, although it does not have cytochrome C oxidase, it does have NADH dehydrogenase, and it also has succinate dehydrogenase. And finally, it has a variety of different terminal electron acceptors that it can utilize, and therefore a variety of different branches that we see in that E. coli ETC. So I actually want to take a moment to look at the E. coli ETC. I don't want you by any means to focus time and energy on memorizing this, but I do want to take a look at it. And after that, I want to look at the oxidase test. And this right here is a picture of the oxidase test. So I want to show you actually a video from Virtual Edge. And so you can be prepping for future labs at the same time as you're learning about how we can assay for cytochrome C oxidase. I'm going to show you that video too.
Good morning. It's very early in the morning for me, so please excuse the sleep and my voice. We're looking here at another great online publication of the Proceedings of the National Academy of Science, or PNAS as we call it. And I really like this particular image because what it shows is the diversity of E. coli's capabilities metabolically. I've joked with some of you in lab that E. coli should really be most fittingly called a flexophile because it is capable of living in all types of different sorts of conditions. And this shows with regards to the presence of oxygen how it can adapt. So notice that on the top portion of this image where this bar is the cytoplasmic membrane of the bacterium, you can see that under aerobic conditions, it has two different branches of its electron transport chain that it can utilize. So this is an NADH dehydrogenase complex. So that is one complex that E. coli has, but then it can use what's called the cytochrome D branch if aeration conditions are low, that is, there isn't very much oxygen present in the environment. This particular component of the ETC will bind very tightly to oxygen and utilize it even if it's present in low concentrations. The cytochrome O branch is going to be utilized when oxygen conditions are high, so when there's a high concentration of oxygen. And then notice too that E. coli doesn't mind when you put it in anaerobic conditions. It just whoop up, shift, and it starts to use its anaerobic branch of the ETC, which it can utilize nitrate as a terminal electron acceptor instead of using oxygen. And you'll notice that nitrate has a pretty high standard reduction potential, so it makes for a pretty good terminal electron acceptor, not as good as oxygen, mind you, but it will utilize nitrate in times of need. And so E. coli just continues to run when it's in anaerobic conditions. And guess what? You've actually seen this up close and personal in lab, because remember when you were looking, those of you who did the oxygen concentration gradient tubes, you did this in lab and you saw that E. coli was, in fact, a facultative anaerobe. So recall how it would grow throughout the tube. So you placed it into this brain-heart infusion. Yes, it really is brain and heart infusion. It's an extract. And you notice that throughout the oxygen gradient, and I can't remember who I was talking to about this, but somebody on the West Bench, right? Troublemakers on the West Bench. You all were asking me about the gradient in the tube. And at the top of the tube, there's high oxygen concentration. So E. coli would be able to use its cytochrome BO branch there, and then it could use its cytochrome BD branch down just a little bit further, because it could still be, the gradient of oxygen is like 100% oxygen at the top, and then it goes down 99, 98, 97, right? And then it continues to drop until somewhere in here it reaches a point that it no longer has oxygen, and then it shifts and it begins utilizing nitrate as a terminal electron acceptor. So while the growth isn't as proliferous, it is still present all the way down to the bottom. So it was hard for me to show in this picture, but you would still be getting some growth all the way down at the bottom. So not only would E. coli grow at the top very proliferously, where it's super glad to have its most preferred terminal electron acceptor, but it can shift and start using nitrate at the bottom of the tube, using its less preferred terminal electron acceptor of nitrate.
As if you don't get enough of me, here we are back in the lab to talk today about the oxidase test. The oxidase test is not a hard test to perform, but it has a couple of little nuances that if you don't do them correctly, then you'll end up not being able to interpret your results. So the first thing that's a little bit tricky about the test is that rather than taking a single isolated colony, as I'm usually harping on you to do, you're going to need to use enough growth on your oxidase test that you can actually see it on the filter paper. So you're actually going to want to pick up quite a lot of growth and put it on your filter paper. So let's start there with some Whatman filter paper. And you just want to place the filter paper into an empty glass petri dish. And then you're going to use a sterile stick, and you're going to take enough growth off of your plate that you can put a little bit onto the Whatman paper and see it on the Whatman paper. Whenever you do this test, you want to have both a positive and a negative control. Your positive control is always going to be Pseudomonas aeruginosa. Pseudomonas will always come up positive because it has that terminal oxidoreductase in the electron transport chain called cytochrome C oxidase, and that's what we're testing for here. So take enough of your Pseudomonas off of your plate that you can see it on your wood stick, and then you should be able to rub it onto your Whatman filter paper such that you can see it on the filter paper as well. If you can't see it, then you don't have enough. You're not going to be able to interpret something that you can't see. That's why this test is so different than any other that we do, because you're actually reading the results right here, right now, and you're not inoculating something and waiting for it to grow. So you're going to want to have always a negative control when you do this test as well. Today we're going to use Shigella dysenteriae as a negative control, but you could use any Enterobacteriaceae. So if you wanted, you could use E. coli or Proteus, or even if you wanted, Yersinia pestis. And again, just take enough that you can actually see it on your stick. And again, rub it onto your filter paper. And you'll want to put it on another corner opposite of where you put your Pseudomonas aeruginosa. Now, when you do this test on your unknown, you're going to want to put your positive control, your negative control, and then you're going to want to put your unknown on the third quadrant of the paper, because this test is extremely time dependent, and so you need to be able to watch the results as they turn. So you would want to always have the three side by side by side. Now we're going to add the oxidase reagent to both the Pseudomonas and the Shigella. The oxidase reagent is a chromatogenic reagent that changes color upon oxidation. So you're just going to want to add a little drop on top of your positive,
And within a few seconds after you add these drops, you're going to want to read the results. So add a drop onto your positive and your negative, and bingo. Look at the dark purple color change on the positive, and the negative just simply stays white. But the longer I sit here and talk and kibitz and whatnot, you're going to watch the negative as it starts to turn purple due to natural oxidation. So time is of the essence. You have to read the results of this test within only a few seconds. If you sit around and let it turn, everything is going to look positive. So it's very important to have a positive and a negative control every time you do this, and to watch the results within only a few seconds after adding the reagent. I hope this gives you the sense that the ETCs of bacterial cells will vary quite greatly. And of course, in Pseudomonas aeruginosa, we see the presence of the cytochrome C oxidase complex, but then remember in E. coli, we did not see that, but we did see NADH dehydrogenase. So as many microorganisms as we have typified, which we know to be very less than 1% of what is out there, we have already seen a great deal of biodiversity within the metabolic pathways and within the electron transport chain. So we're going to shift our attention now to looking at the highly typified mitochondrial electron transport chain. And there are many bacteria who share similarities with the mitochondrial electron transport chain. And so learning it in detail will help us to understand the basic functionings of the chain and to apply it across diverse systems within the bacterial world. Behind me, you'll see a picture of the electron transport chain from Paracoccus denitrificans. You'll notice that it, like E. coli, is capable of using something besides oxygen as a terminal electron acceptor. You can see it reducing nitrate here.
So indeed, calling our attention then to the fact that this is aerobic respiration, so full oxidation utilizing the electron transport chain, everything is running, of glucose. So a whole glucose molecule, looking at the amount of energy yield per glucose molecule. And then lastly, in eukaryotes. So we recognize that this is going to be pertinent. This calculation will be pertinent to the ETC that we just looked at, that we see across the mitochondrial inner membrane. We are going to need the conversion factors that we figured out earlier. So if for each pair of electrons delivered to the ETC by NADH, 3 ATP are formed via OXPHOS. But as we know, the yield is slightly lower for FADH2 or QH2, and we know that that is approximately 2 ATPs. And I should really put approximately here on 3.2. This is a whole number estimate that we use. That is not exactly the amount of ATP yielded, but it gives us an estimate. So using these conversion factors as well as our poster, and of course you've got that out in front of you, I have no doubt. I'm going to also pull up my poster so that we can utilize it. And probably Hannah has already filled in this first box right here for the number of ATP via substrate level phosphorylation from glycolysis. That's a pretty easy one to fill in. And so we will go back to our poster just briefly to make sure of ourselves, recognizing that in glycolysis here in the cytosol of this yeast cell, we would input 1, 2, output 2, that's a 0, but then output 2 more, that's a net of 2. So remember that glycolysis netted 2 ATPs. So we'll go ahead and write that down into this box, recognizing that yield to be 2 ATPs. So I'm going to leave this box blank because it is the most complicated box, and I want to come back to it next. But let's take a look at how many ATPs via substrate level and oxidative phosphorylation for the transition step. So beginning by looking at the transition step, which we see occurring right here in the mitochondrial matrix, notice that it only generates NADH. It doesn't generate any ATP by substrate level phosphorylation, does it? So it's a big whopping 0. So let's put 0 in there for this one. Absolutely no ATP. But of course, that's a horrible 0. But of course, we do notice that it did generate 2 NADHs. And those NADHs are each worth 3 ATPs. So we use this sweet conversion factor from up here. So 3 ATP per NADH. Oh man, I'm already quaking in anticipation of a unit cancellation. Feels so good. So that's going to give us a total of 6 from the transition step. Now let's look at the Tca cycle, because in fact you probably remember that in step 5 of the sweet Tca cycle, there is in fact the generation of an ATP. And remember that for each glucose molecule, the cycle turns not once, but twice. So once would yield 1 ATP, twice would yield 2 ATPs. So there are 2 ATPs made by substrate level phosphorylation. So let's write that down. 2. But we also see the generation of NADHs and FADH2s in the Tca cycle. Remember, reducing power is its middle name. So let's go ahead and look at how many. So 1 turn would yield 1, 2, 3. 2 turns, 4, 5, 6. So 6 NADHs. We can apply our conversion factor again. So good. So we've got 6 NADH times 3 ATP per NADH. Yeah. 18 there. But don't forget the FADH2s. So remember that there's 1 inside of the Wonder Woman enzyme with each turn of the TCA. So 1, 2, there would be 2 FADH2s per glucose. Now, of course, that would be different if we were looking per acetyl-CoA, but we're looking per glucose. So this is going to be 2 FADH2s. Let's apply our conversion factor. So times 2 ATP per FADH2. So we've got our TCA all accounted for. Now we can come back to what I said was the most complicated box. And here's why. Looking at our poster, once again, let's do this. Check out glycolysis and where it's occurring. In a eukaryotic cell, it occurs in the cytoplasm. That means that the NADHs that are generated in the cytoplasm, in order to get their electrons to the electron transport chain, would have to pass the inner mitochondrial membrane. Not happening. NADH, quite simply, does not have the high security clearance that it needs in order to get its electrons across that inner mitochondrial membrane. So the hoopla that it has to go through is to transfer its electrons onto another molecule that has high security clearance. It has to get in there, and then it has to transfer electrons onto NAD+, and then NAD+ can take its electrons to complex 1. So there's a lot of jazz that has to happen to get the reducing equivalence, which is a sexy way to say electrons, from the NADH into the mitochondrial matrix. So that being said, we could pretty well speculate, couldn't we, that this is going to cost some energy. So we can be pretty comfortable with saying that, of course, the 2 NADHs that we just saw being made in glycolysis are not going to be worth the full 3 ATP. And in fact, they're likely to be worth something closer to 2 ATP. But we'll say 2 to 3 to give a range, and note that it may lie in there somewhere, with 3 being an overly optimistic outlook. So we still can go ahead and write down a range here where we can do this quick conversion, where we would recognize that anywhere from 4 to 6 ATPs would be made here. So I put a star on this, and I want to go ahead and write that down so that we make sure we're clear. Depending on how the electrons from NADH enter the mitochondrion, this number varies. So this number varies and could be anywhere from 4 to 6. So that box could be anywhere from 4 to 6. Adding that up, of course, that would be anywhere from 6 to 8 on the total here. So it could be just 4 plus 2 or 6, or it could be 8, way overly optimistic. It's not going to get that high. It's going to be somewhere less than 8. So now we're in a pretty sweet place because we can add everything up. We can go ahead and total for each of the steps in metabolism, but also we can total for the amount made by substrate level versus ox-phos. Now you're starting to see why Sarah in her Baby Got Glucose video said, man, this ox-phos, that's netting a lot of ATP compared to substrate level phosphorylation. So notice at transition step, 3 times 2 or 6. And then 6 times 3 is 18, 2 times 2 is 4. So we're actually looking at counting up to 22. 2 plus 2 is 24. And then we can go ahead and add up in the columns. So 4 via substrate level phosphorylation. How lame does that look compared to ox-phos, where we've got the 22 plus 6 or 28 plus anywhere from 32, ranging from 32 to 34. Wow. Now you can see why Sarah said 32 ATP, right? That's a lot that can come from oxidative phosphorylation, even on the low estimate. So 32 plus 4, or anywhere from 32 to 34 plus 4. So we're looking at 36 to 38, with, of course, that 38 being overly optimistic, but nonetheless getting a whopping tally when we're using both oxidative and substrate level phosphorylation. Now you see why I joked about the yeast giving us the finger when we put the stopper in the bottle, because suddenly they go from generating somewhere around 36 or to 38, or, you know, around in that number, to generating 2. So that is a lot less ATP per glucose. So bear in mind, as I said earlier, that just as the composition of bacterial ETCs varies, so too does the amount of energy yielded. So let's just put energy yield here. So now that we understand the total number of ATP that can come from oxidative versus substrate level phosphorylation, and we see this taking place within the mitochondrion of a eukaryotic cell, this is a really good place to stop for today.